This is Edward Dennis, and I welcome you to this lecture on lipoprotein structure and receptor function. This lecture will be divided into two parts. First is Part A, which will cover structure, and specifically the topics of lipoprotein types and content, the function in metabolism, and hormonal control. And in Part B, we will cover receptors, specifically receptor characteristics and regulation, and then lipoprotein oxidation and atherosclerosis. I have listed here the learning objectives for this lecture on lipoprotein structure and receptor function. I ask that you read these through at your leisure prior to listening to the lecture. On this slide, I have listed the rest of the learning objectives for you to read through at your own pace. Listed here is the reading on which this lecture is based. Lipoproteins are central to the development and progression of atherosclerosis. As leukocytes travel through the arteries, macrophages and other cells accumulate along the wall. And these macrophages take up LDL particles. As they accumulate, one gets fatty streaks as depicted in panel 2, and the formation of foam cells, which are lipid-laden macrophages and other cells. As this process continues, and one gets further accumulation, as shown in panel 3, one progresses to advanced lesions, the formation of a necrotic core, and fibrous cap formation. This eventually leads to rupture, thrombosis, and infarction, as depicted in panel 4. When you get a blood test, the blood that is collected is first spun to remove the red cells. If you take a blood sample just after a meal, the resulting plasma looks as the figure on the right. However, if one has fasted overnight before collecting the blood, then the plasma looks translucent as shown in the panel on the left. Now this difference is due to the lipoproteins. Lipoproteins are really a complex of both proteins, cholesterol, cholesterol esters, triglycerides, and phospholipids. It's a non-covalent complex, and this complex of lipids contains a protein that is termed the apoprotein, which is a specific protein for the particular lipoprotein complex that one is examining. Now, the largest and most prevalent lipoprotein are chylomicrons. These are the largest lipoproteins. They're made mainly of triglycerides, and they are made in the GI tract from the dietary lipids that one has digested and have the function of sending them to the liver. They are marked by a particular apoprotein termed apoprotein B48. The lipoproteins all have generally the same structural features, but vary by composition. As depicted on the right, the central core is an oil consisting of triglycerides and cholesterol esters, and the outer surface is a phospholipid monolayer in which some free cholesterol is embedded. The apoproteins of the lipoprotein are also in or on this surface. Now, the largest lipoprotein after chylomicrons is called very low density lipoprotein, or VLDL, and it is a lipoprotein that is made in the liver and shipped to peripheral tissues. It's marked by its very large size, its abundance of triglycerides, and the ApoB100 protein, which is about twice the size of the apoprotein of chylomicrons. The next smaller lipoprotein is LDL, or low-density lipoprotein, which is derived from VLDL after most of the triglycerides have been removed as it passes through the circulation. The smallest of the lipoproteins is high-density lipoprotein, or HDL, which consists mostly of protein and some cholesterol esters. It's known as the so-called good lipoprotein, and it's marked by apoproteins A and D. Having introduced the various lipoproteins, we can look at their related characteristics. If one starts with chylomicrons and works down to HDL, the density of the lipoprotein is increasing as one goes to the smaller size. This is concomitant with an increase in the protein content. Protein has a density of 1.3, whereas lipids have a density of about 1. So as the protein content increases, the density increases proportionately. The phospholipid content increases somewhat as one goes to the higher density. And the amount of free cholesterol is minimal, but varies somewhat. More striking is the increase in cholesterol esters as one goes from chylomicrons to VLDL to LDL. And the decrease in triglyceride content from 85% in chylomicrons to 50% in VLDL, and then decreases dramatically from there. Another way to think about lipoproteins is simply by size. Chylomicrons are enormous, and that's why when they're present in plasma, they reflect light so dramatically. VLDL are considerably smaller particles, LDL smaller yet, and HDL considerably smaller than the other lipoproteins. The pure protein part of the lipoproteins, the so-called apoproteins, are named in the order somewhat in which they were discovered. So ApoA is in HDL, and there are many forms of ApoA. ApoA1 and ApoA2 particularly prominent. ApoB48, a 240,000 molecular weight protein, is in chylomicrons, whereas ApoB100, about twice the size of ApoB48, is in VLDL, and the resultant LDL that is derived from VLDL. Other apoproteins are distributed among the lipoproteins, I call your attention here particularly to ApoC2, a small apoprotein, but it has a particular activity of activating lipoprotein lipase, and ApoC3, a similarly small apoprotein, inhibits lipoprotein lipase, an important enzyme that we will consider later in this lecture. The chylomicrons are formed in the gut and taken up by the intestine, where they're transported through the lymph or the portal system to the liver. In the process, lipoprotein lipase liberates some free fatty acids from the triglycerides in the chylomicron. The liver produces VLDL, which also, as it passes through the circulation and the capillary endothelial cells, loses its triglyceride by hydrolysis by lipoprotein lipase, and the resulting VLDL remnants are retaken up by the liver. Some of the VLDL loses sufficient triglycerides to become LDL particles, and again, the resultant LDL can be taken up by the liver or by peripheral extrahepatic tissues and cells. HDL precursors are made in the liver as well as the intestine, and these pick up cholesterol from the extrahepatic tissues and result in a process called reverse cholesterol transport, again can end up in the liver. I want to now introduce a number of key enzymes in this process. First of all, the name lecithin is an informal name for phosphatidylcholine. And the enzyme LCAT, which stands for lecithin cholesterol acyltransferase, takes the fatty acid on the middle or SN2 position of phosphatidylcholine or lecithin and transfers it to cholesterol to make a cholesterol ester. This enzyme is activated by another apoprotein, ApoA1. The net effect is that free cholesterol becomes cholesterol ester, and these enzymes, the LCAT enzymes, are found associated with HDL particles and is the key enzyme responsible for the so-called reverse cholesterol transport, because it converts free cholesterol into cholesterol esters on HDL. Another key enzyme is lipoprotein lipase, an enzyme I already introduced. It takes triglycerides and hydrolyzes the fatty acids to make glycerol and 3 free fatty acids. It's located on the surface of the capillary endothelial cells, and it degrades triglycerides in both chylomicrons and VLDLs as they travel by. As I mentioned earlier, it's activated by ApoC2, but inhibited by ApoC3. So you can see another level of control mechanisms that regulate the amount of these large lipoproteins as they travel through the circulation. The final enzyme that I want to introduce is hormone-sensitive lipase. This is an enzyme that resides in the adipose tissue, or our fat cells, and it is activated by protein kinase A, but it works much like lipoprotein lipase, and it provides the mechanism to produce free fatty acids in the adipocytes that can then be taken up by other cells. Those free fatty acids bind to albumin and are transported as such. So this enzyme, which is a lipase-like lipoprotein lipase in that it hydrolyzes fatty acids from triglycerides to make free fatty acids and glycerol. To quickly summarize the scheme, chylomicrons, very rich in triglyceride, are degraded by lipoprotein lipases to make smaller chylomicrons that end up in the liver where the contents are repackaged. The liver creates VLDL and sends it out, and it too is acted upon by lipoprotein lipase to hydrolyze triglycerides, forming some intermediate density lipoproteins denoted here as IDL, and eventually converting them to LDL, or low density lipoproteins, which have much less triglyceride and are retaken up by the liver and repackaged. Some of that LDL travels to peripheral tissues, and it is taken up by a receptor-mediated process in those peripheral tissues, as well as in the liver, which we will describe in more detail in the second part of the lecture. HDL interacts with those peripheral tissues to deliver cholesterol to the liver. At the hormone level, there are two main controls. Insulin, which promotes the storage of fat or lipids, and cyclic AMP, chiefly triggered by epinephrine and glucagon, cause the release of fat or its utilization. Acetyl-CoA carboxylase, which we introduced in the first lecture, is a key enzyme for the synthesis of fat, and it is inhibited by cyclic AMP-dependent phosphorylation, so that one, instead of storing fat, promotes the usage of fat. In contrast, insulin activates the dephosphorylation of acetyl-CoA carboxylase, causing the increased activity of acetyl-CoA carboxylase, and this leads to the promotion of the synthesis of fatty acids through the fatty acid synthase system, and leads to the storage of fat. Now, this is the control in the liver, which is primarily oriented toward biosynthesis of important complex lipids, but also needs energy as well. In the adipocyte, the opposite effect happens. Insulin, which promotes the dephosphorylation of hormone-sensitive lipase, leads to its lack of activity, and therefore one stores more fat, whereas cyclic AMP, which promotes the phosphorylation of hormone-sensitive lipase, leads to its activation and its release of free fatty acids from the triglyceride stored in the adipocyte.